Welcome back to the Deep Dive. Today we are taking you on a journey through, well, an absolute engineering masterpiece. A system that manages to filter, warm, moisturize, and deliver air to the most delicate internal surfaces in the human body.
It's pretty incredible when you stop to think about it.
It is. We are conducting a histological deep dive into the respiratory system, meticulously tracing the path of air, structure by structure, right down to the molecular level. Our mission today is to extract every crucial insight from our source material, providing you with a complete, structured, and highly detailed review.
And it's a crucial system to understand at this microscopic level because its functional demands are just immense. We often simplify it down to just breathing, but really the respiratory system is a comprehensive processing plant.
Processing plant, I like that.
Yeah, and we're going to be tracing that path, following the air as it passes through something like 23 generations of branching passages, and we'll see how the structure changes to meet evolving demands all the way to the tiny air sacs where life support truly happens.
So let's start with the absolute basics. Our source material outlines three principal functions, and I think we all know gas exchange is the star player, but the supporting roles are equally vital.
They are, and that gas exchange, or respiration, is exclusively restricted to the tiniest units, the alveoli. So everything before that has other jobs to do.
So what are those other two main tasks?
That would be air conduction and air filtration. The conduction role is, well, it's handled by that long series of tubes and passages, highways, if you will, that get the air to the gas exchange sites.
Okay, the plumbing.
The plumbing. But without proper filtration, that whole process fails and fast. Filtration is the necessary cleansing, the protection, and the temperature regulation that prepares the inhaled air for the extremely fragile lower respiratory tract.
And beyond those three primary jobs, the system has several essential secondary roles that I think often get overlooked. What else is packed into this apparatus?
Well, there's speech production, which is centered in the larynx. That relies on incredibly precise muscular control and airflow. Then we also have the specialized sense of smell governed by the olfactory mucosa way up in the nasal cavity.
And even smaller roles.
Yes, and they're fundamental. Things like local regulation of immune responses to every single antigen we inhale. And there are even subtle endocrine functions, hormone production and secretion carried out by specialized cells scattered all throughout the lining.
That complexity really makes sense when you consider its developmental history. Before we get into the structures themselves, can you remind us where the lungs and the lower airways actually originate embryologically?
That's a key piece of information for understanding the epithelia, the linings, the entire lower system, the larynx, the trachea, and all the way down to the eventual lung, begins really early in development as a ventral evagination, so essentially an outpocketing of the primitive foregut endoderm.
Okay, so an outpocketing of the gut tube.
Exactly. It's called the laryngotracheal diverticulum.
Yeah.
And because of this origin, the entire epithelial lining of the respiratory system is endodermal.
So the soft functional lining comes from the endoderm. But what about the rigid structures that define the shapes and prevent it from collapsing?
Right, those supportive elements. So the bronchial cartilages, the smooth muscle layers, and the general connective tissue, they all derive from the surrounding embryonic tissue, specifically the thoracic splanchnic mesenchyme. It's a wonderful example of structure and function being defined by the interaction between two different germ layers.
Okay, so now let's look at the grand organizational structure. Our source uses a diagram, figure 19.1, which neatly divides the entire system based on function. How should we visualize that split?
We can trace the air path to see the division really clearly. You have the conducting portion, which is solely dedicated to air transport and conditioning. This path starts at the nasal cavities, goes through the pharynx and larynx, the trachea, the bronchi, and continues all the way down to the terminal bronchiole.
That's a massive tree-like structure.
It is. It involves roughly 23 generations of branching, just a huge amount of plumbing dedicated only to transport.
And after all that,
After that, you hit the respiratory portion. This is the final critical destination. This is where the actual gas exchange happens. It includes the respiratory bronchioles, which kind of serve as a transition zone, then the alveolar ducts, the alveolar sacs, and finally the individual alveoli, which are the terminal respiratory units.
let's focus on the protection necessary for those delicate terminal units. You mentioned air conditioning or climatization, and it's something we often take for granted. Where does the heavy lifting for this process happen?
It's mainly in that conducting portion, and the nasal cavities are by far the most efficient site. The system is designed to achieve three immediate goals. warm the air to a surprisingly high 31 to 34 degrees Celsius.
That high. Wow.
Yeah, and moisten it dramatically to between 90 and 95% relative humidity, and perhaps most critically, remove inhaled particulate matter.
That particle removal sounds like an incredible feat of biological engineering, especially when you consider we inhale thousands of liters of air every day. How effective is this system at filtration, and what are the steps involved?
It's remarkably effective. The process starts right at the entrance with physical filtering by these short, thick hairs called vibrissae, which trap the largest contaminants. Then the mucus and serous secretions coat the walls, and they act like flypaper.
So it's a two-stage trap.
Right, and our sources note that this combined physical and chemical trap successfully eliminates about 95% of all particles with a diameter greater than 15 micrometers.
And once those particles are trapped in that sticky mucus layer, they aren't static. That's where the famed mucociliary escalator takes over.
This is the genius part of the system. The luminal surface of the conducting passages is covered by a continuous sheet of mucus, and beneath that, the specialized epithelial cells are equipped with these synchronized cilia that beat rhythmically, pushing the mucus layer, along with all the trapped debris, continuously up toward the pharynx.
And from there,
Once it reaches the pharynx, it is normally just swallowed. The digestive system's acids then neutralize any contaminants. This sweeping mechanism is absolutely indispensable for preventing contaminants from settling in the lower, sterile parts of the lung.
that provides a perfect transition.
Yeah.
Let's start the structural breakdown right at the entrance. The nasal cavities and sinuses. These are paired chambers separated by the nasal septum, and they're divided into three histologically distinct regions.
Right, so we start with the vestibule, which is the dilated entrance just inside the nostrils. Histologically, it's defined by its lining, which is stratified squamous epithelium, literally a continuation of the skin from your face.
So this is where we find those Vibrissae hairs.
Exactly, and the associated sebaceous glands for that initial entrapment.
And as we move deeper, the epithelium has to adapt from that tough, protective, skin-like lining to the more specialized respiratory lining, right?
Precisely. As we progress posteriorly, the stratified squamous lining thins out and undergoes a distinct transition to the pseudostratified columnar epithelium that characterizes the main respiratory region. This histological shift signals the change from a purely protective barrier to a functional, conditioning surface.
And this respiratory region is the largest section. What are the physical structures that maximize the contact time between the air and the mucosa in this region?
That's the job of the conchae, or turbinates. These are bony, shelf-like projections that protrude from the lateral walls into the nasal cavity. They serve a dual purpose. First, they vastly increase the surface area available for climatization.
More surface area, better conditioning.
Right. But more importantly, they force the airflow into chaotic, swirling patterns. This is a mechanism called turbulent precipitation. This turbulence violently throws suspended particles out of the main airstream and onto the sticky, mucus-covered walls for removal.
So those conchae aren't just increasing surface area, they are purposefully generating turbulence to aid filtration. That's incredible. Now let's drill down into the epithelial cells that line this region, the classic respiratory mucosa. Our source identifies 5 distinct cell types, and they all have very specific jobs.
It's a real team effort. First, you have the ciliated cells. They are the dominant population, making up to 70% of the cells. They are tall, columnar cells, and their cilia are the motors of that mucociliary escalator.
And they need something to push, so next are the goblet cells.
Exactly. The goblet cells make up about 5% to 15% of the population. They are the manufacturers of mucus, synthesizing and secreting the necessary glycoprotein layer that acts as the trap.
Okay, then we have the more specialized players. These seem to handle detection and communication.
Third are the brush cells. These are fascinating. They have short, blunt microvilli on their apical surface, and their basal surfaces are intimately associated with sensory nerve fibers. They function as spiralized chemosensory receptor cells.
So they're tasting the air, in a way.
In a way, yes. They monitor the chemical quality of the air and mediate general sensations and protective reflexes, like triggering a sneeze.
And the fourth type fits into the diffuse neuroendocrine system.
Those are the small granule cells, also known as Kolchitsky cells. They belong to the DNES, the body's scattered endocrine system. They contain numerous small secretory granules packed with hormones, which can include things like serotonin or various polypeptides like bombesin. They're thought to contribute to local regulation, perhaps sensing changes in the air composition or pressure.
And finally, number 5, the essential stem cell reserve.
The basal cells. They're small cells resting directly on the basement membrane, but they don't extend up to the lumen. They are the critical stem cell pool, acting as progenitors for all the other cell types, allowing for continuous epithelial renewal and repair.
So below that complex epithelium is the lamina propria, and this is where the air warming happens. You described it earlier as a biological heat exchange system. What makes the vascular network so effective at this job?
Well, the lamina propria is just extremely rich in vasculature. It forms a highly complex network of superficial capillary loops, and the design here is brilliant. The blood flow in these loops is arranged perpendicular or countercurrent to the direction of the inhaled airflow.
Countercurrent. Why is that important?
This countercurrent arrangement maximizes the thermal gradient. It ensures maximum heat transfer from the circulating blood to the inhaled air, effectively warming it to body temperature very efficiently before it ever reaches the trachea.
It really is like a sophisticated radiator built right into our nose.
Yeah.
And if we get an infection or an allergy, why does that efficient vascular system lead to that classic congestion?
It's a direct result of that design.
Yeah.
When you have inflammation, whether from a virus or an allergic response, the capillaries in the lamina propria become engorged or leaky. This vascular swelling and the accumulation of fluid edema causes the lamina propria to expand dramatically, which severely restricts the narrow air passage created by the conchae.
And that physical restriction is what we feel is nasal congestion.
That's exactly it.
Speaking of reflexes, the mechanical process of the sneeze, stimulated by those sensory brush cells, is shockingly powerful.
It's a rapid-fire defense mechanism. A particle stimulates the sensory nerves, which relays a signal to the brainstem. The body then executes a rapid, deep inspiration. Then, structures in the larynx, the vocal folds, vestibular folds, and the epiglottis slam shut,
Trapping the air.
trapping the air and allowing immense pressure to build up from the contracting diaphragm and thoracic muscles. When these structures suddenly open, the air, liquid, and trapped debris are expelled at extraordinary velocities, up to 50 meters per second. That's about 180 kilometers per hour.
Wow. That sheer speed is necessary to forcefully clear the nasal passages.
It is. We also need to remember that the respiratory mucosa is an immunological battlefield, constantly dealing with antigens. The lamina propria is highly cellular, housing significant populations of lymphocytes, including specialized gamma delta T cells, as well as macrophages and mast cells. This highly active local lymphatic tissue provides immediate immune surveillance.
Okay, let's shift gears to the specialized olfactory region. This is the area dedicated to the sense of smell, located in the upper third or apex of the nasal cavity.
Yes, and it's a small, specialized patch, maybe about 10 square centimeters in humans. It's visibly identifiable as slightly yellowish-brown due to the presence of pigment. Histologically, it's very distinct because, although it is pseudostratified epithelium, it completely lacks goblet cells.
No goblet cells. What does it have instead?
It has 4 unique cell populations, which are defined visually in diagrams like figure 19.3.
Let's break down those specialized cells, starting with the heart of the system, the neuron.
The key functional cell here is the olfactory receptor cell. It's a bipolar neuron that spans the entire thickness of the epithelium. And this is one of the most remarkable facts in neurobiology. These are some of the only neurons in the postnatal body that are regularly replaced.
They're constantly regenerated.
Constantly regenerated by basal cells with a lifespan of only about 1 month. It's amazing.
So how does this specialized neuron capture the odorant molecules?
Its apical domain is structurally modified into an olfactory vesicle, and projecting radially from that vesicle are about 10 to 23 long non-modal cilia. These cilia, which lie flat along the epithelial surface, are the sensory antennas. This is where the specialized G protein receptors, the Golf receptors, are housed.
And the supporting cast for these neurons?
The most numerous cells are the supporting cells, or sustentacular cells. They are tall columnar cells, and one of the easiest ways to identify the olfactory region in light microscopy is that their nuclei are positioned more apically, so closer to the surface, than the nuclei of the receptor cells.
And they do more than just support, right?
Oh, absolutely. They provide mechanical and metabolic support, but critically, they synthesize the odorant-binding proteins, OBPs, which are essential for the process.
And the other cells?
You have the basal cells, which are the stem cells for both the receptor neurons and the supporting cells. And we still find a few brush cells acting as general chemosensors, just like they did in the respiratory region.
Okay, so describe the full path of the signal, the olfactory transduction pathway, which is laid out in figure 19.4. It involves multiple steps to convert a chemical binding into an electrical impulse.
It starts outside the cell. Odorant molecules have to first dissolve in the olfactory mucus. They then bind specifically to those OBPs that are secreted by the supporting cells. The OBP acts like a shuttle.
A delivery service.
Delivery service, exactly. It delivers the odorant to the surface of the cilia. There, the odorant binds to its specific G protein-coupled receptor, the Golf receptor, and that initiates the cascade.
And the cascade is what converts the chemical signal into electrical energy.
Correct. The G protein activation kicks off the CAMP cascade within the cell. This cascade ultimately leads to the opening of ion channels, specifically sodium and calcium channels. The subsequent influx of ions causes a depolarization of the neuronal membrane, which generates an action potential that shoots down the axon.
It's such a sophisticated system, especially considering the sheer number of scents we can distinguish.
It's a remarkable coding system. Humans possess over 350 different types of olfactory receptors, and we're able to perceive thousands of distinct odors through a process called population coding. The brain doesn't just recognize one receptor firing, it interprets the unique pattern of activity across that entire population of over 350 receptor types.
So what happens when the signal leaves the epithelium? Where does it go?
The basal domains of the receptor neurons give rise to unmyelinated axons. These axons collect into bundles that form the olfactory nerve, which is cranial nerve height. They then pass through the tiny holes of the cribriform plate to reach the olfactory bulb in the brain.
And this structure is fragile.
Very fragile. Severe trauma to the head can shear those axons right where they pass through the bone, leading to permanent anosmia or loss of smell.
The function of this whole system relies heavily on the Bowman glands. What do these glands provide that's so important?
These are characteristic branch tubulo-alveolar serous glands located in the lamina propria, just beneath the epithelium. Their secretion is watery and protein-rich, and it is absolutely vital for two functions. First, it prevents the fragile cilia from dehydrating, which is essential for maintaining the ion gradients required for depolarization.
And the second function?
The constant flow of this watery secretion acts as a continuous solvent and flush for odorants. This ensures that once an odor has been detected, it is quickly washed away, making the receptors immediately ready to detect the next incoming odorant. It's a reset mechanism.
Now, connecting this detailed histology to modern pathology, the anosmia seen in COVID-19 patients. The mechanism our sources describe is quite surprising because the neuron isn't the primary target.
It is surprising. Studies show that the SARS-CoV-2 virus did not directly infect the olfactory receptor neurons. Instead, the virus targets the adjacent supporting syntaccular cells.
So it's the support cells that get hit.
Exactly. These supporting cells are the ones that express high levels of the viral entry proteins, ACE2 and TMPRSS2. When they get infected, the supporting cells are destroyed, causing temporary damage and dysfunction to the neighboring receptor neurons. But because the receptor neurons are constantly regenerated by basal cells, and the supporting cell population eventually recovers, often within 10 days, the sense of smell is usually restored.
So that explains the characteristic transient anosmia. It's damage to the infrastructure, not the core sensory element. Okay, moving briefly to the paranasal sinuses. What are these extensions and what is their role?
The sinuses, the ethmoid, frontal, sphenoid, and maxillary, are essentially air-filled extensions of the respiratory region, and they are lined by a thin respiratory epithelium. Their functions include adding resonance to the voice. Their thin mucus layer is continuously swept by cilia into the main nasal cavities.
But the sinuses have an important secret function related to a highly reactive gas.
That's nitric oxide, or NO. The paranasal sinuses are the major source of NO production within the entire airway system, resulting in concentrations several times higher than elsewhere.
And why is that beneficial?
For two reasons. In the nasal cavity, it acts as a local defense mechanism against invading microbes. But once it's inhaled and reaches the lungs, NO is a critical molecule that helps facilitate gas exchange and acts as a potent vasodilator, relaxing vascular smooth muscle and thereby reducing pulmonary vascular resistance.
So a protective mechanism in the nose and a functional regulator in the lung. Now for the clinical view from folder 19.1, what does rhinitis look like histologically?
Rhinitis, the common cold is simple inflammation. Microscopically, the key features are marked tissue swelling or edema in the lamina propria. This is due to vascular engorgement and fluid leakage, and it's accompanied by a heavy infiltration of inflammatory cells. If that inflammation spreads into the sinuses, it becomes rhinosinusitis.
And allergic rhinitis, or hay fever, has a very distinct histological signature tied to the immune response.
Yeah, it's defined by the allergen binding to IgE antibodies that are sitting on mast cells in the lamina propria. This binding triggers mast cell degranulation, releasing potent chemical mediators like histamine and leukotrienes.
Which cause all the classic symptoms.
Exactly. Increased secretion, rhinorrhea, and congestion. Critically, if it becomes chronic, the histology shows two telltale signs: a thickening of the basement membrane and a massive infiltration of specialized inflammatory cells, eosinophils, and mast cells into the lamina propria.
That concludes the initial filtration phase. Once the air is cleaned and conditioned, it needs a robust highway to the lungs. That brings us deeper into the conducting system: the pharynx, larynx, and trachea.
The pharynx is the junction box connecting the nasal and oral cavities to the lower systems, the larynx for air, the esophagus for food. It's also a key resonating chamber for speech. And we should note the presence of significant lymphatic tissue there, like the pharyngeal tonsil and its connection to the middle ear via the eustachian tubes.
Next, the larynx, the organ of phonation. This is structurally complex, supported by a mix of hyaline and elastic cartilages like the thyroid, cricoid, and the epiglottis.
Internally, the mucosa forms two key pairs of folds separated by the ventricle. The upper pair are the vestibular folds, often called false vocal cords. They are immobile and function primarily in resonance and protecting the airway entrance.
And the lower pair are the true engines of sound, the vocal folds, which delineate the opening called the rima glottis.
Exactly. These folds contain the vocal ligament and, most importantly, the vocalis muscle, which is skeletal muscle. The mechanism of phonation is the controlled vibration of these folds as expelled air passes over them.
And pitch is controlled by tension.
Pitch is determined by the tension applied by intrinsic laryngeal muscles. Stretched or tense folds produce higher frequencies and a higher pitch, while relaxed folds produce a deeper pitch.
Let's examine the epithelial lining of the larynx, which is shown in figure 19.6. We see that familiar adaptation to mechanical stress again.
We do. Anywhere there is intense abrasion from fast-moving air or potential contact with food. So, specifically, the luminal surface of the vocal cords and the superior surface of the epiglottis. The lining is a robust stratified squamous epithelium, or SSE.
Then the rest.
The rest of the larynx, where the physical stress is lower, is lined by the standard ciliated pseudostratified columnar epithelium for sweeping debris up and out.
Our sources also point out a clinically relevant space within the vocal fold.
Yes, the Ranke space. This is a layer of connective tissue situated directly beneath the stratified squamous epithelium of the vocal cord. It's clinically significant because it is poorly vascularized and notably lacks lymphatic vessels. Pathology, like fluid accumulation or tumor development, can occur specifically within this distinct space.
Moving down, we reach the trachea, the main conducting pipe. It's a relatively short, flexible tube, about 10 to 12 centimeters long, positioned right in front of the esophagus, what defines its structure and prevents collapse.
The trachea's defining characteristic is the support provided by a series of 16 to 20 incomplete, C-shaped rings of hyaline cartilage. These rigid rings keep the lumen open at all times.
C-shaped, not complete rings. So what bridges the posterior gap of those C rings?
That gap, which is right up against the esophagus, is bridged by fibroelastic tissue and the trachealis muscle, which is smooth muscle. Contraction of this muscle helps regulate the diameter of the tracheal lumen, especially during a forceful expulsion of air, like during a cough.
Okay, now for the 4 layers of the tracheal wall, which are shown in figure 19.7.
From the lumen outward, they are: 1, the mucosa, which is the epithelium and lamina propria; 2, the submucosa, which is denser connective tissue containing glands; 3, the cartilaginous layer with the C-shaped hyaline rings; and 4, the adventitia, which is the outer connective tissue that binds the trachea to adjacent structures.
Let's look closer at the tracheal epithelium in figures 19.8 and 19.9. It's the classic respiratory epithelium again, featuring the same 5 cell types we discussed earlier. The ciliated cells are the most abundant, powering the escalator.
And the ciliated cells are absolutely vital here. Each one bears around 250 cilia, and their precise, synchronized beating is the physical engine of the mucociliary escalator, constantly sweeping contaminants up to the pharynx. The other cell types, goblet cells, basal cells, brush cells, they all perform the same maintenance and sensory roles.
Let's discuss those specialized neuroendocrine cells again, the small granule cells. What is their potential role here in the trachea?
The small granule cells are still part of the DNES here, found singly near the basement membrane. They secrete hormones like serotonin, embomasin, or catecholamines. Sometimes they cluster together with sensory nerve endings to form structures called neuroepithelial bodies.
And the function of those bodies.
Well, it's hypothesized that these structures act as chemoreceptors or baroreceptors, helping the body reflexively regulate the caliber of the airways or the local blood vessels in response to subtle changes in oxygen levels or air pressure.
Okay, so below the epithelium, the text describes a distinctive histological feature, what appears to be an unusually thick basement membrane. What is this layer actually composed of?
In a standard H&E stain, it looks like a thick, glassy, homogeneous line. But electron microscopy reveals that it's not the basal lamina itself, but an unusually dense and thick reticular lamina, which is the deepest part of the lamina propria. It's composed of densely packed collagenous fibers.
And this thickening is a significant histological marker.
It is. The text specifically notes that this layer becomes thicker and more pronounced in individuals suffering from chronic irritation, like heavy smokers or asthmatics, as shown in folder 19.3.
So where does the mucosa end and the submucosa begin?
The boundary between the lamina propria and the submucosa is theoretically defined by a distinct, though sometimes hard to see, elastic membrane. The lamina propria itself is loose cellular connective tissue packed with immune cells, lymphocytes, plasma cells, mast cells forming what's called the bronchus-associated lymphatic tissue, or BALT.
A localized immune defense post.
That's a good way to put it. Then the submucosa is a denser layer of connective tissue, housing larger blood vessels and, importantly, the submucosal glands. These glands are typically mucus acini, often capped with serous demilunes, and they deliver a high volume of glycoproteins to the surface, serving as a critical backup source of mucus.
We have to touch upon the crucial clinical correlation here from folder 19.2. Squamous metaplasia. Why does the body perform this conversion, and why is it so dangerous?
Metaplasia is a reversible adaptive change. The delicate, high-maintenance, ciliated, pseudostratified columnar epithelium gets replaced by a much tougher, multi-layered stratified squamous epithelium, SSE. This happens normally in areas of chronic physical stress, like on the vocal cords, but pathologically it's a key feature in chronic bronchitis and, notably, in smokers.
So why does smoking trigger this change? What's the mechanism?
The noxious elements in smoke directly impair the function of the cilia, essentially stalling the mucociliary escalator, so the body has to compensate for the inability to clear mucus by frequent, forceful coughing.
And that chronic stress causes the change.
That chronic mechanical stress, combined with the chemical irritation, forces the lining to swap its ciliated cells for the tougher SSE, which is much more resistant to abrasion. The danger is that if the irritation, the smoking, isn't removed, this protective but abnormal SSE has a high potential for malignant transformation,
Which is the precursor to.
squamous cell carcinoma, a very common form of lung cancer.
That structural change really highlights how the body prioritizes immediate survival, like physical toughness over long-term function, like mucus clearing. Okay, let's now follow the air as it branches into the progressively smaller airways, bronchi and bronchioles.
The trachea bifurcates into the two main or primary bronchi. The right bronchus is anatomically wider and shorter, which is why inhaled foreign objects tend to lodge there. These primary bronchi enter the hilum and divide into the lobar secondary, bronchi 3, for the 3 lobes of the right lung and 2 for the 2 lobes of the left.
And those split again into the segmental tertiary bronchi,
Oh.
which define key surgical units.
Exactly. 10 on the right, 8 on the left. Each segmental bronchus supplies a self-contained unit known as a bronchopulmonary segment. And our source emphasizes these segments because they are physically separated by connective tissue septa and have their own blood supply, making them convenient, isolated units for surgical resection if a disease is localized.
What are the two key histological changes that define the transition from the trachea to the main intrapulmonary bronchi?
The structures change drastically. First, the C-shaped rings of hyaline cartilage are replaced by irregular cartilage plates that completely surround the circumference of the airway. This changes the bronchial shape from D-shaped to circular.
And the second change?
A complete continuous layer of smooth muscle, known as the muscularis, is introduced, wrapping entirely around the tube.
So the addition of that complete muscularis layer means the bronchial wall now consists of 5 distinct layers.
Yes. Mucosa, muscularis, submucosa, a cartilage layer of discontinuous plates, and the outer adventitia.
Okay, but why does the body replace the strong, consistent C-rings with these irregular cartilage plates? Isn't stability still key this deep in the lung?
That's a great question. Stability is key, but so is flexibility and maneuverability within the lung tissue itself. The C-rings are great for preventing tracheal collapse, but they don't allow for much dynamic size adjustment. The plates still prevent the tube from fully collapsing, but that surrounding smooth muscle layer allows for much finer dynamic control of the airway diameter.
Which is crucial for regulating airflow.
Crucial for regulating airflow distribution throughout all the lung lobes.
We now move to the bronchioles. Structurally, they are defined by what they lose.
They are defined by the definitive absence of cartilage plates and submucosal glands. They're much smaller, generally 1 millimeter or less in diameter, and they represent the final segment of the purely conducting system before gas exchange begins.
The source also gives us some terminology for the structural units supplied by these small bronchioles.
Right. A pulmonary lobule is supplied by one bronchiole. A smaller unit, the pulmonary sinus, consists of the final conducting tube, the terminal bronchiole, and all the respiratory bronchioles and alveoli that it aerates. And the smallest functional unit is the respiratory bronchiolar unit, just a single respiratory bronchiole and its attached alveoli.
Let's trace the epithelial transformation through the bronchioles.
It's a rapid transformation. The large bronchioles start with ciliated pseudostratified epithelium, which quickly transitions to simple ciliated columnar. As the airways narrow further toward the terminal bronchioles, the lining becomes simple cuboidal epithelium.
And a key cell disappears.
Critically, yes. The mucus-producing goblet cells, which were so prevalent upstream, disappear entirely in the smaller bronchioles.
So if the goblet cells disappear, how does the system protect itself and prevent the walls from sticking together?
That's where a new specialized cell takes over. The club cells, which were previously known as Clara cells. These are non-ciliated, dome-shaped secretory cells that increase in number as you go deeper, essentially replacing the protective function of the goblet cells.
Tell us more about the club cell function, shown in figure 19.14. What does that distinct dome shape and internal structure reveal?
Their structure is highly indicative of their function. They have basal rough ER and an abundance of apical smooth ER and secretory vesicles. They produce and secrete a vital surface active agent, which is a lipoprotein.
A kind of surfactant.
Exactly, a miniature surfactant. It coats the internal walls of these tiny bronchioles to prevent their walls from adhering to each other and collapsing during exhalation. This is essential for keeping the smallest conducting airways open. They also produce club cell secretory protein, CC16, which is a measurable biomarker for pulmonary health.
The transition zone, then, is the respiratory bronchiole. This is where we see the first instances of gas exchange.
Yes, they are very narrow airways, lined mostly by simple cuboidal epithelium, a mix of club cells and a few ciliated cells. Their distinguishing feature is the presence of scattered, thin-walled outpocketings extending from their lumen. These are the first alveoli, marking the point where the conducting system ends and the respiratory system begins its primary job.
Let's look at two major diseases that target these smaller conducting airways. First, folder 19.3, asthma.
Asthma is a chronic inflammatory condition causing recurrent reversible airflow obstruction. The obstruction is due to intense inflammation and bronchospasm, the aggressive, uncontrolled constriction of that thick layer of smooth muscle in the bronchioles.
What are the key microscopic changes we would identify in an asthmatic airway wall?
You'd see a profoundly inflamed wall, heavily infiltrated by characteristic immune cells, including eosinophils, T cells, and mast cells. The epithelium is often damaged and shows an increase in goblet cells leading to excess mucus.
And structural changes.
Most critically, two structural elements are thickened. The basement membrane is noticeably pronounced due to collagen deposition, and the smooth muscle layer itself is thickened, a process called hyperplasia, which makes the airway hyperreactive to stimuli.
The second major pathology from folder 19.4 is cystic fibrosis, CF, which directly involves a failure of cellular transport in the epithelium.
CF is a severe autosomal recessive disorder resulting from a mutation in the CFTR gene, which codes for a crucial chloride ion channel. The defect in this channel causes a catastrophic failure of ion transport. Specifically, there is decreased chloride secretion into the lumen and, concurrently, an increased reabsorption of sodium ions and water out of the lumen.
And the result of pulling that water out of the airway surface fluid.
The mucus layer becomes profoundly dehydrated, sticky, unusually thick, and highly viscous. This thick, gummy mucus cannot be cleared by the cilia.
So the mucociliary escalator just gets completely stuck.
It malfunctions and clogs completely. This persistent blockage leads to chronic infection, inflammation, and eventual severe structural damage, defining CF as a chronic obstructive pulmonary disease.
That brings us to the ultimate destination of the air pathway, the alveoli, and the marvel of the air-blood barrier.
The alveoli are the terminal air sacs, the site where all gas exchange occurs. They're small polyhedral chambers, only about 0.2 millimeters across. But while they're small individually, their collective internal surface area is staggering about 75 square meters in total.
Which is roughly the size of a single tennis court.
Exactly. That vast surface is what allows for instantaneous gas transfer.
The air flows from respiratory bronchioles through alveolar ducts, which are like elongated hallways defined by alveoli lining their periphery, and into alveolar sacs, which are clusters of alveoli at the end of a duct.
And separating every adjacent alveolus is the interalveolar septum, the thin connective tissue core that holds the dense network of pulmonary capillaries. This septum is the foundation of the air-blood barrier.
Let's define the cell types that form this delicate alveolar epithelium.
There are two main players. First, the type I alveolar cells, or type I pneumocytes. These are squamous cells, meaning they are extremely flat and thin, and they cover about 95% of the alveolar surface area. They are joined by powerful occluding junctions, creating a tight, impermeable barrier.
And they can't divide.
Right, they are terminally differentiated, meaning they cannot divide or repair themselves.
So if they cover 95% of the surface, what are the type 2 alveolar cells doing?
The type 2 pneumocytes are cuboidal cells, and they tend to cluster at the junctions where the septa meet. They only cover about 5% of the surface, but they make up about 60% of the total alveolar cell population. They are the secretory powerhouses and the regenerative backbone of the alveolus.
What is their defining internal structure, as seen in figure 19.17?
Their cytoplasm is filled with these characteristic concentric layers of lipid and protein called lamellar bodies. These bodies are storage and delivery vesicles for surfactant, the critical surface active agent that type 2 cells synthesize and secrete.
And why are type 2 cells the crucial backbone of the alveolus?
They function as the progenitor cells for both type I and type 2 cells. When the delicate type I cells are damaged or destroyed, the type 2 cells proliferate, differentiate into new type I cells to repair the barrier, and replenish the type 2 population. Seeing type 2 cell hyperplasia in a biopsy is a clear histological indicator of alveolar injury and repair.
The function of surfactant is essential for life. What is its core mechanism, and what is its most critical component?
Surfactant's primary job is to dramatically reduce the surface tension at the air-epithelium interface. Without it, the cohesive forces of the fluid lining the alveoli would be too strong, causing the alveoli to collapse completely like a deflated balloon upon exhalation.
And the key chemical component.
The single most important chemical component responsible for this tension reduction is the phospholipid dipalmitoyl phosphatidylcholine, or DPPC.
This immediately links us to neonatal respiratory distress syndrome, RDS.
RDS is a life-threatening condition for preterm infants, typically born before the 35th week of gestation. At this early stage, the type 2 pneumocytes have not matured enough to produce sufficient quantities of functional surfactant,
leading to widespread alveolar collapse.
Yes. Fortunately, it is often treated effectively today, either by administering exogenous surfactant directly into the lung or by giving the mother antenatal corticosteroids to accelerate the fetal type 2 cell maturation.
And beyond DPPC, the surfactant layer includes several specialized proteins, SPA, B, C, and D, that have roles beyond just physical mechanics.
Right. SPB and SPC are necessary for organizing and stabilizing the DPPC layer. But SPA and SPD are highly important for host defense. They're part of the innate immune system, capable of binding to microorganisms and modulating local immune responses to keep the lower airways sterile.
Let's now visualize the ultimate structure, the air-blood barrier, as shown in figures 19.20 and 19.21. This is the physical distance across which oxygen and CO2 must diffuse. The source describes a thin portion and a thick portion.
The thin portion is the highly optimized segment where the fastest gas exchange occurs. It consists of only 4 extremely thin layers. Starting from the airspace, you have 1, the thin film of surfactant, 2, the cytoplasm and basal lamina of the type I epithelial cell.
And then the key architectural feature.
The fused basal lamina. It's shared by both the epithelial cell and the endothelial cell. And then layer 4 is the cytoplasm of the capillary endothelial cell. That fusion of the two basal laminae is what minimizes the diffusion distance.
And the purpose of the thick portion.
In the thick portion, the basal laminae are separated by a small interstitial space containing connective tissue, elastic fibers, and various septal cells. This area is critical for maintaining fluid balance. Tissue fluid that leaks into the interstitium accumulates here before being drained away by nearby lymphatics.
If that delicate barrier is compromised, say during severe pneumonia, what does the resultant tissue damage look like?
Well, acute interstitial pneumonia or other forms of severe lung injury can destroy the type I cells or the capillary endothelial cells. When that barrier is breached, plasma proteins leak uncontrollably into the alveolar space.
And that leakage is what causes the real problem.
Exactly. When these exuded proteins mix with the components of surfactant, they coagulate to form dense, homogeneous, eosinophilic structures called hyaline membranes that line the alveolar walls. This severe pathology, known as diffuse alveolar damage, essentially creates a thick, impenetrable layer that blocks gas exchange.
We have to mention the alveolar macrophages, the crucial defense mechanism inside the air spaces.
These are the dedicated cleanup crew. They're derived from blood monocytes, and they are constantly patrolling the alveolar airspace to scavenge and remove inhaled particulates, which earns them the descriptive name dust cells.
And there's a distinct clinical use for these macrophages, shown in figure 19.22.
Yes, the heart failure cells. In cases of severe left ventricular failure, blood backs up in the lungs, causing pulmonary congestion and sometimes tiny hemorrhages into the alveoli. The alveolar macrophages phagocytize these leaked red blood cells.
And that leaves behind a telltale sign.
As the RBCs break down, they leave behind the iron-rich brown pigment called hemosiderin. Macrophages engorged with this brown hemosiderin are specific markers of chronic heart failure.
There are also macrophages that reside permanently in the septa, unlike the migratory dust cells.
Those are the septal macrophages. They live within the connective tissue of the interalveolar septa, and they are known for retaining phagocytized materials, particularly inert substances like inhaled carbon particles or silica, sometimes for the entire life of the individual.
Finally, what mechanism ensures that if one airway branch gets blocked, the distal alveoli can still receive air?
That's the system of collateral air circulation, mediated by the alveolar pores of Kohn. These are small openings found in the interalveolar septa that allow air to pass directly from one alveolus to an adjacent one. In obstructive lung disease, where a bronchiole might be completely blocked by mucus, these pores become critically important for aerating the obstructed region from neighboring healthy areas.
Let's move into our final section, integrating these histological findings with major systemic diseases and the vascular supply. Starting with folder 19.5, covering chronic obstructive pulmonary disease, COPD.
COPD is an umbrella term including conditions like chronic bronchitis, severe asthma, and emphysema. And emphysema is histologically defined by the permanent enlargement of the air spaces distal to the terminal bronchiole, which is accompanied by the irreversible destruction of the alveolar wall.
The destruction of that alveolar wall, the septum, is the key mechanism of function loss.
Absolutely. The loss of septal walls means a catastrophic reduction in the total surface area available for gas exchange. The most common cause is chronic inhalation of irritants, overwhelmingly cigarette smoking.
And the pathology is linked to an imbalance of enzymes.
It is. Smoking triggers the release of large amounts of destructive proteases, particularly elastase from neutrophils and macrophages. This elastase excessively breaks down the elastic components of the septal wall.
And there is a genetic link as well.
Yes. A genetic deficiency in the enzyme inhibitor alpha-1 antitrypsin results in severe, often early-onset emphysema. Alpha-1 antitrypsin is normally responsible for neutralizing those destructive proteases. Without it, the elastase runs unchecked, accelerating the destruction of the alveolar walls significantly.
Next, pneumonia, the general inflammation of the lung tissue, usually infectious. Pathologically, the sources detail distinct stages of progression.
In bacterial pneumonia, we see a clear progression. The initial acute phase involves massive fluid and immune cell influx. The first main stage is called red hepatization, where the alveoli are filled with protein-rich exudate, vast numbers of neutrophils, and red blood cells. The lung tissue becomes heavy, firm, and takes on a liver-like consistency.
Hence the term hepatization.
Exactly. This is followed by the gray hepatization stage, where congestion diminishes, and the red cells have been lysed and absorbed, leaving primarily a mass of fibrin and neutrophils. Eventually, macrophages arrive to clean up the debris, allowing the tissue to return to normal function.
The damage caused by SARS-CoV-2 COVID-19 infection targets specific cell types, leading to a distinct pathology.
COVID-19 pathology focuses on cells expressing the ACE2 receptor, which are primarily the type 2 pneumocytes and endothelial cells. The resulting damage is that severe diffuse alveolar damage we talked about, characterized by heavy edema, the formation of those telltale hyaline membranes, massive proliferation of type 2 pneumocytes trying to repair the tissue, and extensive vascular damage, often accompanied by microthrombosis.
Finally, let's wrap up with the systemic support. Blood supply, lymphatics, and nerves. The lungs operate with a crucial dual circulation.
This is a really important distinction. The pulmonary circulation, derived from the pulmonary artery of the right ventricle, is dedicated solely to gas exchange. It follows the bronchial tree, supplying the capillaries of the interalveolar septa for oxygenation, and its venous drainage returns the oxygenated blood directly to the left atrium.
And the second system, the nutritional supply.
That's the bronchial circulation, derived from the bronchial arteries, which branch off the aorta. This is the systemic circulation, and it supplies the metabolic needs of virtually all lung tissue except the alveoli themselves, so the walls of the bronchi, the connective tissue, and so on.
And these two systems connect.
They do. They anastomose or connect at the junction between the conducting and respiratory passages, and most of the blood delivered by the bronchial circulation ends up draining back into the pulmonary veins, mixing with the highly oxygenated blood just before it enters the left atrium.
And the lymphatics also utilize a dual drainage system.
Correct. One set of lymphatic vessels drains the parenchyma of the lung, following the air passages back toward the hilum. The second set drains the lung surface, traveling in the visceral pleura. Both sets ultimately converge at the hilum, passing through several groups of lymph nodes, the bronchopulmonary and tracheobronchial nodes, which serve as collection and immune filtration centers.
Oh, finally, the innervation, which regulates the dynamic function of the airways.
The innervation is primarily controlled by the autonomic nervous system, involving both sympathetic and parasympathetic divisions. These nerves mediate reflexes that modify the diameter of the air passages by regulating smooth muscle contraction, particularly in the bronchioles, and they also govern the rate of glandular secretion from the respiratory mucosa.
What a deeply integrated and responsive system. We trace the journey from the robust initial defenses of the stratified squamous epithelium and the ingenious filtration systems of the nasal cavity powered by that mucociliary escalator. We followed the structural transitions in the bronchi, where the cartilage support changed to accommodate the continuous dynamic smooth muscle layer.
And we culminated in the alveoli, understanding the absolute necessity of the progenitor type 2 cells to produce surfactant and repair the ultra-thin, four-layered air-blood barrier, which is constantly under threat from infection and environmental insults. Every step, every cell type, from the DNES cells possibly sensing air quality to the club cells protecting the smallest airways, serves a specific, critical purpose.
What stands out most to me is that capacity for self-renewal, even in traditionally non-regenerative tissues. We noted the constant replacement of olfactory neurons and the progenitor capability of type 2 alveolar cells, which is essentially the lung's native stem cell repair crew.
Indeed. And it raises a compelling question that researchers are grappling with right now. Given that the type 2 alveolar cells possess such robust progenitor capability, capable of restoring both type I and type 2 populations after acute injury, how much further might regenerative medicine advance lung repair?
So, could we harness this inherent capacity for renewal?
Exactly. Could we harness it perhaps through targeted growth factors or cellular therapies to eventually bypass the need for organ transplants in devastating diseases currently defined by irreversible structural damage, like advanced severe emphysema?
A fascinating area for future exploration. If we can leverage the lung's own resilience, the treatment landscape for chronic respiratory illness could be fundamentally transformed. Thank you for joining us on this comprehensive deep dive into the histology of the respiratory system.
A pleasure.
We encourage you to continue tracing the connections in your own learning journey.